So when I think about the intestinal microbiome, and that's where most of what I'm going to focus on in this talk is the intestine, I think of it in three compartments: the epithelium, the immune system, and the microbiome. And on this Venn diagram, you can see they all touch each part and have an overlap. So one part we oftentimes think of in isolation, but they truly all impact each other. And part of some of the therapeutics as we go forward, at least some of the investigation into them, play that similar role. So as we've already heard, the intestinal microbiome for each person is individual, and it functions in its own homeostasis. There's the lining, the immune cells, the bacteria that are in the lumen. But in the setting of critical illness, there is a dysbiosis or a pathobiome that's class starting to be described, where we have intestinal epithelial injury, a change in the type of bacteria that are still present, a virulence change in these bacteria that are there, immune system dysfunction that allows for these bacteria to potentially invade the host. The idea of what we would like to get back to is return this patient into a setting of homeostasis. So some of the potential therapeutic avenues that are under investigation are some of the things I'll talk about: prebiotics, probiotics, fecal microbial transplant, something that's a little bit older that has some mixed data, selective digestive tract decontamination. And then I just have some even further therapeutics to talk about that are sort of pipe dreams. So starting with the prebiotics, they really function as these non-absorbable particles that can be given into the gut, into any patient truly. The most specific of which that has a lot of data coming out around it is fiber. Fiber can be split into two different categories: fermentable and non-fermentable. And these are some examples of the fermentable fiber that we could eat in a diet. Non-fermentable fiber has some data coming out, but most of what we talk about here are the fermentable type of fibers. And the importance about the administration of the fiber is what they allow the bacteria that are present to do. They're able to produce short-chain fatty acids, two of which most important being acetic acid or acetate and butyrate, because a lot of the signaling pathways that the immune cells and the epithelium use come from these bacteria to get back into that homeostatic picture. So some of this, the data that's come out about this, looking at short-chain fatty acids in relation to intestinal barrier function. In this study, animals were given LPS as sort of their model for sepsis. And you can see that the intestinal permeability went up, but when they gave the short-chain fatty acids, in this case was butyrate, the permeability went down. And they also looked at the tight junction proteins and noticed that there was recovery in the tight junction proteins ZO1, occludin, claudin-1. Claudin-2 is a different type of protein and acts the opposite of those three, so it was down-regulated as appropriate. But you can see giving the short-chain fatty acid improved the intestinal epithelial barrier. And we know that barrier function is dysregulated in the setting of sepsis and is associated with a worsened mortality in animal models. The other aspect, as I mentioned, there's this Venn diagram that dietary fiber, when it's converted to these short-chain fatty acids, acts as a signaling molecule to immune cells, specifically regulatory T cells that express FOXP3 that are able to dampen down inflammation to reduce any further intestinal epithelial damage, but also uses the signal to stimulate IgA production in the B cells. IgA, as we know, is secreted across luminal barriers and help regulate the types of bacteria that are present in the gut and prevent pathogenic bacteria from overgrowing. So in this study, animals were given this acetate as a short chain fatty acid, and showed that there was an improvement in their IgA response targeted to the microbiota that was in the gut lumen. These GPR43 knockout animals are animals that don't respond to acetate. So you can see that the expression of their IgA decreased compared to their wild type controls, because they don't have the ability to receive that signal. And whenever for the follow-up within this, they measured the IgA in the stool and saw that when they gave to wild type animals completely, when they gave acetate, they had an appropriate response in increasing their IgA targeted at the specific microbiome bacteria that were present. If we take this a little bit more into the clinical realm, there have been some small studies that are sort of pilot trials looking at the administration of fiber. In this sort of mixed heterogeneous ICU small 20-patient pilot study, we saw that there was an increase in the abundance of bacteria that were present in the patients who received fiber compared to those who did not receive fiber. And at the same time, they measured short chain fatty acid concentration, and they noticed that though not statistically significant, trended towards significance in this small study. More ends may have made this more significant, so this is promising and at least sort of a pilot to get things going. The other avenue being probiotics. I know that someone's going to talk about probiotics specifically in their talk, so I won't go too much into it, but I can certainly answer any questions about it as well. But probiotics are basically a group of commensal bacteria that are felt to benefit the gut and systemic health. Most of the studies have been positive. They've been small, but the problem with some of the protocols that have been used have been issues with standardization. Most of the studies don't use the same bacteria. They don't use the same route of administration, orally versus via endoscopy versus rectal. We don't have a baseline flora, just like we mentioned. Everyone is an individual, so we don't know where they started from and how they moved and what that means for everybody. And then the timing, are they getting how long after sepsis, immediately when they're diagnosed, further on? It was very variable, so it's hard to interpret some of those results. Furthermore, most of these patients are getting concomitant antibiotics for the treatment of their sepsis, which can reduce any probiotic that's being induced to promote healthy changes. So it's a little bit of a mixed picture and needs to sort of be parsed out in the future. There's certainly been concern when patients are immunocompromised at baseline or from their critical illness as to giving them bacteria, if that's going to be harmful or not. And there certainly have been some reports of systemic colonization into the bloodstream of some of the probiotics that were given in some of these patients. So these aren't things that we should take seriously and be smart before we start using them as actual treatment therapies. One of the other promising avenues is fecal microbial transplant. This is taking donor stool that is felt to have a healthy microbiome, good alpha diversity, which is a lot of different bacteria within itself, and good beta diversity compared to someone else, which is the type of bacteria that are there compared to another patient's bacteria, and giving it to someone who's an unhealthy recipient. And it's already being used as part of the algorithm for recurrent C. difficile colitis infections, but hasn't really translated over into the sepsis realm very much. We have some preclinical data that looked very promising from Dr. Alberti's lab at the University of Chicago. They use a model of surgery plus sepsis, where they do a partial hepatectomy and then give these animals a human pathogenic slurry into their cecum. And you can see that if they have just surgery, they all survive. But if they get this slurry, the black line goes down, their survival is significantly lower than the just surgery. With these animals, what they did was they gave them back a fecal microbial transplant, and it recovered and rescued their sepsis mortality up to the blue line here. Their control for that was an autoclaved fecal transplant to say that the bacteria and what was in the stool is what was actually causing it. And they also measured the butyrate and the short chain fatty acid in these animals and showed that the fecal transplant recovered and rescued the effect of the short chain fatty acid. So there have been some usage of fecal transplant in the ICU for critically ill patients, most of which have been limited to case reports and small studies. So this is a list of most of them that have been used outside of the setting of Clostridium difficile infection. But I want to bring your attention to this. Most of these patients were much further out from their initial septic insult, except for the last patient. It was still 7 days, but that's also still further out from the initial presentation. So some caution needs to be in line with this, especially as what I mentioned before about immunocompromised states, concomitant antibiotics, things like that, before moving towards fecal transplant. Selective gut decontamination, as opposed to adding bacteria back and promoting good health, the idea from this was attempt to remove the bad bacteria, usually and classically through non-absorbable enteral antibiotics. There have been mixed results. There have been some promising data earlier, but some of the newer meta-analyses don't really show a lot of resistance, promise, especially if there is high resistant bacteria present at the time of decontamination. And hasn't been widely used across ICUs because of the concern for inducing further resistance with leaving certain bacteria behind. So just briefly on the future therapies, things that have been discussed in the literature about bacteriophage engineering to attempt to change bacterial function, bacterial editing themselves, altering the intestinal barrier, making the tight junction stronger or weaker depending on their function, using the intestines as part of an immunization and inducing IgA to sort of prime the immune system to be ready in case there is a systemic infection. And then just the metagenome and what these bacteria do to the system and how the host responds and being able to control that influence.